Hello friend, and welcome to Science for Sleep. Did you know that plants are constantly sending and receiving information, even though they have no brain, no nerves, and no sound? Every plant you have ever seen is quietly processing signals from its environment, responding through chemistry rather than thought, and coordinating its own survival moment by moment. Tonight, we're going to begin by gently rethinking what a plant actually is. For most of us, plants fade easily into the background of daily life. They line sidewalks, fill parks, sit on windowsills, and appear in photographs as scenery rather than subjects. Because they do not move quickly, speak audibly, or react in obvious ways, it's easy to assume that plants are mostly passive, present, but not actively doing much at all. In reality, plants are living systems operating continuously. Unlike animals, plants do not rely on rapid motion or centralized control. They have no brain issuing commands, no nervous system transmitting electrical impulses across long distances in milliseconds. Instead, plants function through distributed processes. Each part of the plant, roots, stems, leaves, and even individual cells, plays a role in sensing, responding, and adjusting to changing conditions. Plants are always balancing trade-offs. They regulate how much water to lose and how much carbon dioxide to take in. They adjust growth direction based on light and gravity. They modify internal chemistry in response to temperature, damage, or the presence of other organisms. None of this happens all at once, and none of it happens quickly by human standards. But it happens continuously. This slowness is not a limitation. It is a strategy. Plants evolved in environments where staying rooted was not a disadvantage, but a necessity. Rather than escaping threats through movement, plants developed ways to endure them. Over time, this led to systems that emphasize efficiency, patience, and long-term stability, rather than speed or reaction. Because of this, plant activity often goes unnoticed. Their movements are measured in hours or days instead of seconds. Their responses unfold gradually, blending into the background rhythm of the environment. To a human observer, a plant may appear unchanged from morning to night, even though internally it has shifted water, redistributed energy, adjusted chemical concentrations, and responded to subtle cues in light and air. Plants are not inert objects waiting to be acted upon. They are active participants in their surroundings. A plant does not simply sit in sunlight. It measures light intensity, direction, and duration. It alters leaf position, internal chemistry, and growth patterns in response. A plant does not merely absorb water. It controls uptake carefully, managing pressure within cells to maintain structure and prevent damage. A plant does not passively exist alongside other plants. It releases chemicals into the air and soil, influencing nearby organisms in ways that science is still working to fully understand. When we describe plants as living systems, we are recognizing that life does not require speed, noise, or intention, as humans define it. Life can also be quiet, distributed, and persistent. It can operate without a center, without urgency, and without rest in the way we usually imagine it. This perspective allows us to move away from seeing plants as decoration or backdrop. Instead, we can begin to see them as companions, organisms sharing time and space with us, unfolding their lives at a different pace according to different rules. Throughout this video, we'll explore plants from the inside out. We'll look at how they are built, how they move water and energy, how they communicate without sound, and how they form partnerships with other living things. We'll take our time, moving slowly through these systems, just as plants themselves do. For now, it's enough to remember this. Every plant you encounter is alive in ways that are easy to overlook, quietly sensing, adjusting, and responding, moment after moment, without ever needing to be noticed. And with that understanding, we can begin to look closer. To understand how plants function as living systems, it helps to begin at the smallest scale where life is organized: the cell. Every plant, no matter how large or long-lived, is built from microscopic units that carry out the work of survival. These plant cells are not simple or uniform. They are highly structured environments, each one maintaining balance, pressure, and chemical activity at all times. One of the most defining features of a plant cell is its cell wall. Unlike animal cells, which are enclosed only by flexible membranes, plant cells are surrounded by a rigid outer layer made primarily of cellulose. This wall provides structural support, protects the cell from mechanical stress, and helps maintain shape. Because of cell walls, plant tissues can form upright stems and broad leaves without the need for bones or internal skeletons. Inside the cell wall sits the cell membrane, which carefully regulates what enters and leaves the cell. Water, ions, sugars, and signaling molecules pass through this membrane in controlled ways. Even at this microscopic level, plant life depends on balance rather than force. Movement across membranes is often gradual, governed by concentration differences and specialized transport proteins, rather than rapid bursts of activity. One of the most prominent structures inside many plant cells is the central vacuole. This large, fluid-filled compartment can occupy up to 90% of the cell's interior. The vacuole serves several functions at once. It stores water, dissolved nutrients, and waste products. It helps regulate internal pressure, known as turgor pressure, which keeps plant tissues firm and upright. It also plays a role in detoxifying harmful substances and maintaining internal chemical balance. Turgor pressure is especially important for understanding how plants hold their shape. When water fills the vacuole, it presses outward against the cell wall. The wall resists this pressure, creating a state of tension that keeps leaves firm and stems supported. When water is scarce, turgor pressure drops and plants begin to wilt. This visible change reflects a shift happening at the cellular level, repeated millions of times across the plant's tissues. Another defining feature of plant cells is the presence of chloroplasts. These specialized organelles contain chlorophyll, the pigment responsible for capturing light energy. Chloroplasts are the sites of photosynthesis, where light energy is converted into chemical energy stored in sugars. This process does not happen instantly. It unfolds through a series of tightly regulated chemical reactions, each step depending on the structure and organization of the cell. Chloroplasts are not evenly distributed throughout the plant. They are most concentrated in leaf cells, where light exposure is greatest. Their position within cells can even shift slightly in response to light intensity, helping protect the cell from damage during periods of excessive brightness. This kind of adjustment happens silently, without movement visible to the naked eye, but it reflects a constant internal awareness of environmental conditions. Plant cells also contain mitochondria, the same energy-releasing structures found in animal cells. While chloroplasts build sugars, mitochondria break them down through respiration to release usable energy. This means that plant cells, like animal cells, are always managing both energy production and energy consumption. Even in darkness, when photosynthesis stops, respiration continues. Beyond these major structures, plant cells include networks of membranes, enzymes, and scaffolding proteins that keep internal processes organized. The cytoskeleton helps position organelles and guide growth. The endoplasmic reticulum and Golgi apparatus assemble and transport proteins and carbohydrates. Each component contributes to the smooth, ongoing operation of the cell as a self-contained system. What makes plant cells especially remarkable is how they cooperate without central command. No single cell controls the plant. Instead, millions or billions of cells respond locally to their own conditions, guided by chemical signals and physical constraints. Out of these countless small interactions emerges the stable form of a leaf, the steady rise of a stem, or the slow expansion of a root. Seen this way, a plant is not a single object, but a vast community of cells, each one maintaining pressure, managing resources, and responding to signals in its immediate environment. Life at this scale is quiet and continuous. There are no sudden starts or stops, only ongoing adjustment. We'll build on this cellular foundation to see how plant cells organize into tissues and how those tissues work together to create the larger structures we recognize as roots, stems, and leaves. For now, it's enough to remember that every visible part of a plant rests on an invisible world of cells, each one steadily doing its work. Once we understand the individual plant cell as a living, self-regulating unit, the next step is to see how countless cells organize themselves into larger structures. A single plant cell can maintain balance and carry out basic life processes, but it is the cooperation between cells that allows a plant to grow tall, spread wide, and persist for years or even centuries. Plants do not develop through centralized planning. There is no command center directing construction. Instead, organization emerges gradually as cells divide, specialize, and settle into positions where their structure and chemistry suit a particular role. Over time, this leads to the formation of tissues, groups of cells that share similar functions and work together to support the plant as a whole. One of the simplest but most important plant tissues is the epidermis. This outer layer of cells covers leaves, stems, and roots, forming a boundary between the plant and its environment. Epidermal cells are tightly packed, creating a protective surface that reduces water loss and limits damage. In many above-ground tissues, the epidermis is coated with a waxy layer known as the cuticle. This thin film helps prevent dehydration, while still allowing light to pass through to the cells beneath. Embedded within the epidermis are specialized structures such as stomata and hair-like projections called trichomes. Stomata allow controlled gas exchange, while trichomes can reduce water loss, reflect excess light, or deter herbivores. These features illustrate how even a thin outer layer can perform multiple functions at once, balancing protection with exchange. Beneath the epidermis lies ground tissue, which makes up much of the plant's internal volume. Ground tissue is responsible for a variety of tasks, including photosynthesis, storage, and structural support. In leaves, ground tissue cells contain abundant chloroplasts and form the primary site of photosynthesis. In roots and stems, these cells often store carbohydrates and other resources, acting as reserves the plant can draw upon during periods of growth or stress. Ground tissue also contributes to flexibility and strength. The arrangement of cells and the thickness of their walls allow tissues to bend without breaking. This is especially important in environments where wind, rain, or passing animals apply constant mechanical forces. The ability to flex rather than resist outright helps plants avoid damage while remaining upright. Perhaps the most distinctive tissues in plants are the vascular tissues, which form the plant's internal transport network. Vascular tissue is divided into two main types: xylem and phloem. Xylem conducts water and dissolved minerals from roots upward through the plant. Phloem distributes sugars and other organic compounds from areas of production to areas of use or storage. Together, these tissues allow distant parts of the plant to remain connected despite their physical separation. Vascular tissues are arranged in continuous strands that extend from the tips of roots to the edges of leaves. This continuity is crucial. Water absorbed deep in the soil can reach the highest leaves of a tree, while sugars produced in sunlit leaves can be transported to underground roots. The plant functions as an integrated system because these tissues maintain open pathways over long distances. As tissues form, they are organized into larger structures known as organs. Roots, stems, and leaves are not separate inventions, but coordinated outcomes of tissue arrangement. Each organ contains all three major tissue types: epidermal, ground, and vascular, configured in ways that suit its specific role. A root's tissues prioritize absorption and anchoring. A leaf's tissues maximize surface area and exchange. A stem's tissues emphasize support and transport. What is striking about this organization is its flexibility. Plants do not produce a fixed body plan at birth. They continue generating new tissues and organs throughout their lives. Buds can form into branches or flowers. Roots can extend, branch, or thicken in response to soil conditions. This open-ended development allows plants to adapt continuously to their surroundings. Throughout this process, communication between cells remains local and chemical. Hormones, pressure gradients, and nutrient flows guide development without instructions written in advance. Cells respond to their immediate neighbors and internal states, and the larger pattern emerges gradually over time. By viewing plants as systems built from tissues rather than as static objects, we begin to see how structure and function are inseparable. Each layer supports the next. Each tissue contributes to the whole, and the plant persists not because of rapid action, but because of steady coordination. With this understanding of tissues and systems in place, we can now move outward, beginning with the hidden half of the plant, the roots, and explore how they anchor, absorb, and support everything that grows above the surface. When we think about plants, our attention usually goes upward, towards stems, leaves, and flowers. But much of a plant's life takes place below the surface, out of sight. Roots are not simply anchors holding a plant in place. They are active, responsive systems that manage water, nutrients, stability, and communication with the surrounding environment. At their most basic level, roots serve as anchors. By growing downward and outward into soil, roots secure the plant against gravity, wind, and movement above ground. This anchoring function becomes especially important for larger plants and trees, where the weight and surface area of the upper structure place constant mechanical stress on the base. A stable root system distributes these forces through the soil, reducing the risk of toppling. But anchoring is only one part of what roots do. Roots are also the primary interface between the plant and the soil. They absorb water and dissolve minerals that are essential for growth, metabolism, and structural support. This absorption does not occur evenly across the entire root surface. Instead, it is concentrated in fine extensions called root hairs. These microscopic projections dramatically increase surface area, allowing roots to make close contact with soil particles and the thin films of water that surround them. Water enters root cells through osmosis, moving from areas of higher water concentration in the soil to lower concentration inside the plant. Minerals such as potassium, nitrate, and phosphate often require active transport, meaning the plant expends energy to move them into root cells against concentration gradients. Even underground, where nothing appears to be happening, roots are constantly managing flows of water and ions. Roots are also sensory organs. They detect gradients in moisture, nutrient availability, oxygen levels, and even soil density. When water is more abundant in one direction, roots tend to grow toward it, a response known as hydrotropism. In compacted or oxygen-poor soil, root growth may slow or redirect. These adjustments happen gradually, guided by local conditions rather than centralized instructions. Different plants develop different root architectures depending on their environment and strategy. Some plants produce deep taproots that penetrate far below the surface, accessing water reserves unavailable to shallow-rooted neighbors. Others form wide, fibrous root systems that spread horizontally near the surface, quickly absorbing rainfall before it drains away. Both strategies reflect adaptations to climate, soil type, and competition. Beyond absorption and anchoring, roots often function as storage organs. Many plants store carbohydrates, primarily in the form of starch, within their roots. These reserves can be mobilized later to support new growth, flowering, or recovery after damage. In this way, roots act as long-term energy banks, buffering the plant against fluctuations in light and seasonal change. Roots also play a central role in the plant's interactions with other organisms. The soil surrounding roots, known as the rhizosphere, is one of the most biologically active zones on Earth. Roots release sugars, amino acids, and other compounds into the soil, shaping the microbial communities that live nearby. These microbes, in turn, influence nutrient availability, disease resistance, and overall plant health. Some roots form close partnerships with fungi, creating mycorrhizal associations that extend the plant's effective reach through the soil. Others host nitrogen-fixing bacteria that convert atmospheric nitrogen into forms the plant can use. These relationships will be explored in more detail later, but it's important to recognize that roots are rarely working alone. Unlike animal organs, roots are not fixed in number or shape. Plants continuously produce new roots while older ones age and decay. This ongoing turnover allows the plant to adjust its underground presence as conditions change. After a dry period, new roots may grow deeper. After disturbance near the surface, growth may shift outward or downward. All of this activity unfolds without sound or sudden movement. Roots grow millimeter by millimeter, responding to subtle differences in pressure, moisture, and chemistry. Their work is steady and patient, shaping the plant's relationship with the soil over long stretches of time. By the time we notice a plant standing upright and stable above ground, its root system has already performed countless quiet adjustments. It has secured the plant, gathered resources, stored energy, and negotiated with the living soil around it. The visible plant depends entirely on this hidden half, working continuously below the surface. With roots established as the foundation of plant life, we can now follow the path water takes as it moves upward, traveling from soil through roots and into the internal transport systems that support everything above ground. Once water enters a plant through its roots, it begins a journey that can span remarkable distances. In a small plant, that journey may be only a few centimeters. In a tall tree, it can extend tens of meters from soil to the highest leaves. This upward movement happens continuously, without pumps, muscles, or moving parts, relying instead on the physical properties of water and the structure of plant tissues. The pathway that carries water upward is known as the xylem. Xylem tissue forms long, hollow channels that run from roots through stems and into leaves. These channels are made from specialized cells that, once mature, are no longer alive. Their walls are thickened with compounds such as lignin, which provide strength and prevent collapse under tension. By forming continuous tubes, xylem allows water to move efficiently over long distances. Water enters the xylem from root tissues after passing through several layers of cells. Along the way, the plant carefully regulates which minerals accompany the water, maintaining internal balance. Once water reaches the xylem, it becomes part of a connected column extending upward through the plant. What drives this upward movement is not pressure from below, but pull from above. As water evaporates from the surfaces of leaves, it creates tension within the xylem. This process, known as transpiration, removes individual water molecules from leaf tissues and releases them into the air as vapor. When one molecule leaves, it pulls on the molecule behind it, which pulls on the next, and so on, all the way down the column to the roots. This chain-like movement is possible because water molecules are strongly attracted to one another. The cohesion between water molecules allows them to remain connected, forming a continuous column even under tension. At the same time, water molecules adhere to the walls of the xylem, helping stabilize the column and prevent it from breaking. Together, these forces, cohesion and adhesion, allow plants to lift water against gravity without expending metabolic energy on pumping. The energy driving this movement ultimately comes from the sun, which powers evaporation at the leaf surface. The strength of this system is evident in tall trees. Despite their height, they are able to transport water from deep roots to leaves exposed to sun and wind. Under normal conditions, this transport operates smoothly and silently. However, the system is not without limits. Extreme drought or freezing temperatures can introduce air bubbles into the xylem, disrupting the continuity of the water column. Plants have evolved various strategies to reduce or repair such disruptions, including producing new xylem tissue over time. Water moving through the xylem does more than hydrate cells. It carries dissolved minerals essential for plant function. Nutrients such as calcium, magnesium, and nitrate travel upward with the water, reaching leaves and growing tissues where they are incorporated into cellular structures and metabolic processes. The flow of water also helps regulate temperature. As water evaporates from leaves, it removes heat, cooling plant tissues much like perspiration cools human skin. This cooling effect can be especially important during warm, sunny conditions, helping prevent damage to delicate cellular machinery. What makes the xylem system particularly suited to plant life is its steadiness. Water movement is not sudden or intermittent. It rises continuously, adjusting to environmental conditions such as humidity, temperature, and light intensity. On dry, hot days, transpiration increases, pulling water upward more rapidly. On cool or humid days, the pull weakens and flow slows. Throughout this process, the plant remains responsive without being reactive. There is no single switch that turns water flow on or off. Instead, countless tiny adjustments at the leaf surface collectively influence the movement of water through the entire plant. By following water's upward journey, we begin to see how plants rely on physical principles as much as biological ones. Their internal systems are shaped to work with gravity, evaporation, and molecular attraction rather than against them. In the next section, we'll turn our attention to the downward and sideways movement of sugars through the plant, tracing how energy captured in the leaves is distributed to support roots, stems, and future growth. As water rises from the roots and moves upward through the plant, it requires a stable pathway. That pathway is provided by the stem. Stems are often thought of simply as connectors linking roots to leaves, but their role is far more substantial. They provide structure, support, transport, and long-term stability, allowing plants to grow upward and outward while remaining grounded. At a basic level, stems hold leaves in positions where they can access light. By elevating leaves above the ground and spacing them apart, stems reduce shading and improve exposure to sunlight and air. This spatial arrangement is not random. Over time, stem length, branching patterns, and angles adjust to environmental conditions, shaping how the plant interacts with its surroundings. Inside the stem runs the plant's internal transport system. Vascular bundles, made up of xylem and phloem, form organized pathways that move water, minerals, sugars, and chemical signals throughout the plant. These bundles are arranged differently, depending on the type of plant. In many flowering plants, they form a ring, while in grasses, they are scattered. Regardless of arrangement, their function remains the same, maintaining continuity between distant parts of the plant. In plants with soft, flexible stems, known as herbaceous plants, rigidity comes primarily from water. As water fills the cells of the stem, turgor pressure builds, pressing outward against cell walls. This internal pressure gives the stem firmness, allowing it to stand upright without hard, woody tissue. When water availability decreases, turgor pressure drops, and stems may bend or wilt. This change reflects a shift at the cellular level rather than structural failure. Woody plants rely on a different strategy. In trees and shrubs, stems undergo secondary growth, gradually increasing in thickness over time. This growth is driven by a thin layer of dividing cells called the vascular cambium, which produces new xylem on the inside and new phloem on the outside. As layers accumulate year after year, the stem becomes stronger and more resistant to bending and breakage. The xylem produced during secondary growth forms what we commonly recognize as wood. These xylem cells are reinforced with lignin, a complex polymer that adds rigidity and durability. Once formed, these cells no longer transport water actively, but remain as part of the plant's structural framework, supporting weight and resisting environmental stress. Over time, this process leaves behind visible records of growth. In many climates, trees produce annual growth rings as the rate of cambium activity changes with the seasons. Rings formed during periods of rapid growth differ in density from those formed during slower periods. By examining these rings, scientists can infer past conditions such as rainfall, temperature, and even disturbances like fire or drought. In this way, stems quietly archive the history of the environments in which plants live. Beyond support and transport, stems also serve as sites of storage and renewal. Some plants store carbohydrates or water in their stems, using these reserves to survive unfavorable conditions. Others produce buds along the stem, containing dormant tissues that can later develop into branches, leaves, or flowers. This allows plants to grow modularly, adding new parts while older sections remain functional. Stems must also remain flexible enough to withstand movement. Wind, rain, and the weight of snow apply shifting forces that could cause rigid structures to snap. The internal arrangement of fibers and tissues within stems allows them to bend slightly and return to shape, distributing stress rather than resisting it outright. This balance between strength and flexibility is essential for long-term survival. Unlike animal skeletons, plant stems are not constructed all at once. They are built gradually, layer by layer, responding to conditions as they grow. Strength accumulates where it is needed, and form emerges over time rather than being predetermined. This slow construction allows plants to remain upright without bones, relying instead on water, chemistry, and careful organization. With stems providing support and transport, we can now turn to the structures they hold aloft. In the next section, we'll look closely at leaves, not as simple green surfaces, but as complex exchange zones where light, air, and energy move continuously between the plant and its environment. Leaves are often the most recognizable part of a plant. Their shapes, colors, and textures vary widely, and they are frequently treated as decorative features rather than functional ones. In reality, leaves are carefully engineered exchange surfaces. Their primary role is not appearance, but interaction, with light, air, water, and energy moving constantly through and across them. One of the defining characteristics of a leaf is its large surface area relative to its thickness. By spreading outward into thin, flat forms, leaves maximize exposure to sunlight. This design allows light to penetrate leaf tissues efficiently, reaching the chloroplast where energy capture takes place. A thick, compact structure would limit this access, reducing the plant's ability to gather energy from its environment. Inside the leaf, the internal structure is arranged to support exchange. Just beneath the upper surface lies a layer of tightly packed cells rich in chloroplasts. These cells absorb light and carry out most of the plant's photosynthesis. Below them is a region of loosely arranged cells with many air spaces between them. These spaces allow gases such as carbon dioxide and oxygen to diffuse easily throughout the leaf. Gas exchange is a continuous process. Carbon dioxide from the air enters the leaf through small openings, moves through the internal air spaces, and reaches photosynthetic cells. Oxygen produced during photosynthesis follows the reverse path, diffusing back into the atmosphere. This movement occurs passively, driven by concentration differences rather than active transport. Veins run through the leaf like a branching network. These veins are composed of xylem and phloem, the same vascular tissues found in stems. Xylem delivers water and dissolved minerals to the leaf, replacing water lost through evaporation. Phloem carries sugars produced in the leaf to other parts of the plant, where they can be used for growth, storage, or repair. The close spacing of veins ensures that no part of the leaf is far from a transport pathway. Leaf shape plays a significant role in regulating exchange. Broad leaves capture more light, but may lose water more quickly. Narrow or divided leaves reduce water loss and heat buildup, which can be advantageous in dry or hot environments. Some leaves have waxy surfaces that limit evaporation, while others are covered in fine hairs that reflect sunlight or trap moisture near the surface. Leaves are also sensitive to orientation. Many plants adjust the angle of their leaves throughout the day, reducing exposure during intense midday sunlight and increasing it when light levels are lower. These movements are often slow and subtle, driven by changes in water pressure within specialized cells at the base of the leaf or leaf stalk. Over time, these adjustments help balance energy gain with water conservation. Despite their importance, leaves are not permanent structures. They are built to function for a limited period and are regularly replaced. In many plants, leaves are shed seasonally in response to changes in temperature or light availability. This shedding reduces water loss and prevents damage during unfavorable conditions. In other plants, individual leaves age and fall away gradually as new ones form. The temporary nature of leaves reflects an efficient use of resources. Rather than investing heavily in long-lasting structures, plants produce leaves that perform their task and are then recycled. Nutrients are often withdrawn from aging leaves and redistributed before they are shed, minimizing waste and supporting new growth elsewhere. Leaves also play a role in temperature regulation. As water evaporates from leaf surfaces, it removes heat, cooling the tissue. This cooling effect helps maintain stable internal conditions, protecting sensitive cellular machinery from overheating. In this way, leaves contribute not only to energy capture, but also to environmental control within the plant. Seen as exchange surfaces rather than ornaments, leaves reveal the quiet complexity of plant life. They are places where light becomes chemical energy, where gases move invisibly, and where water and nutrients flow in steady balance. Their design reflects the plant's need to interact constantly with its surroundings while remaining rooted in place. With leaves established as the primary sites of exchange, we can now look more closely at the process they support most directly. Soon, we'll explore photosynthesis as a daily routine, a repeating sequence of reactions that quietly powers nearly all life on Earth. Photosynthesis is often described as a dramatic transformation, light becoming life in a single moment. In reality, it is better understood as a steady, repeating routine. For plants, photosynthesis is not an event, but a continuous maintenance process, unfolding quietly as long as conditions allow. At the center of this process are chlorophyll molecules housed within chloroplasts. When light reaches a leaf, photons interact with these pigments, exciting electrons and raising them to higher energy states. This excitation does not create sugar directly. Instead, it sets in motion a chain of reactions that temporarily store energy in chemical forms the plant can use. These early reactions depend on light and occur within specialized membranes inside the chloroplast. Water molecules are split during this stage, releasing oxygen as a byproduct. The oxygen diffuses out of the leaf and into the atmosphere, while the energized electrons and chemical carriers remain within the plant, ready for the next phase. The second phase of photosynthesis does not require light directly, but it relies on the energy captured earlier. During this phase, carbon dioxide from the air is incorporated into organic molecules through a series of enzyme-driven reactions. Over multiple steps, small carbon-based compounds are assembled into sugars. This process is precise and regulated, ensuring that energy is stored efficiently rather than released all at once. The sugars produced through photosynthesis serve several purposes. Some are used immediately as fuel for cellular respiration, supporting ongoing metabolic needs. Others are converted into starch and stored within plant tissues, creating reserves that can be drawn upon later. Still others are transported to growing regions such as roots, developing leaves, or reproductive structures. Photosynthesis does not operate at a fixed rate. Its pace shifts continuously in response to environmental conditions. Light intensity, temperature, water availability, and carbon dioxide concentration all influence how efficiently energy is captured and stored. On bright, well-watered days, photosynthesis may proceed steadily. Under cloudy skies or drought conditions, it slows, conserving resources until conditions improve. This flexibility is essential for survival. Plants cannot relocate when conditions change, so their internal processes must adjust instead. Enzymes involved in photosynthesis are sensitive to temperature, functioning best within certain ranges. Water availability influences both the chemical reactions themselves and the opening of stomata, which regulate carbon dioxide intake. Each adjustment helps maintain balance rather than maximizing output. Photosynthesis also follows daily rhythms. In most plants, activity increases after sunrise, reaches a plateau during the day, and tapers off as light fades. These patterns are influenced not only by immediate conditions, but also by internal biological clocks that help plants anticipate regular changes in their environment. Even when light levels fluctuate, internal rhythms provide a framework for timing energy capture. What makes photosynthesis especially suited to plant life is its integration with other systems. Water transport through xylem supplies the raw material for early reactions. Stomatal regulation balances carbon dioxide intake with water loss. Sugar transport through phloem distributes the products of photosynthesis to where they are needed. Each system supports the others, forming a coordinated whole. Rather than producing bursts of energy, photosynthesis spreads energy capture across time. This steady approach reduces stress on cellular systems and aligns with the slow growth patterns of plants. Energy is accumulated gradually, stored, shared, and reused, supporting life without urgency. When viewed as a routine rather than a spectacle, photosynthesis becomes easier to appreciate. It is a quiet process, repeating day after day, leaf by leaf, sustaining plants and shaping the atmosphere around them. Through this steady exchange between light, air, and chemistry, plants maintain themselves and, in doing so, support countless other forms of life. While photosynthesis builds energy-rich molecules, plants also rely on a second, equally important process: respiration. Respiration allows plants to access the energy stored in sugars, releasing it in controlled amounts to power growth, repair, and daily maintenance. This process operates continuously, regardless of whether it is day or night. Respiration begins with sugars produced during photosynthesis or drawn from stored reserves. These sugars are broken down through a sequence of chemical reactions inside plant cells. As bonds are rearranged and broken, energy is released and captured in usable forms that the cell can immediately apply to its needs. This energy fuels everything from cell division to the movement of molecules across membranes. Unlike photosynthesis, respiration does not require light. It occurs in the mitochondria of plant cells, the same organelles responsible for respiration in animals and fungi. Oxygen is consumed during this process, and carbon dioxide is released as a byproduct. This means that plants, like animals, are constantly using oxygen to sustain their internal chemistry. Because respiration never stops, plants continue consuming oxygen at night when photosynthesis has paused. This often leads to the misconception that plants steal oxygen in darkness. In reality, the amount of oxygen used by plants is small compared to the total available in the air, and the oxygen released during daylight far outweighs nighttime consumption. The balance between these two processes depends on light conditions and overall plant activity. The rate of respiration varies throughout the plant. Actively growing tissues, such as root tips, young leaves, and developing buds, respire more rapidly than older, fully formed tissues. These growing regions require large amounts of energy to build new cells and structures. In contrast, mature tissues primarily use energy for maintenance, repair, and regulation. Temperature plays a significant role in respiration. As temperatures rise within a suitable range, chemical reactions speed up, increasing the rate at which sugars are broken down. When temperatures drop, respiration slows. This sensitivity to temperature helps explain why plants grow more quickly during warm seasons and enter periods of reduced activity during cold conditions. Respiration also adjusts in response to stress. During drought, limited water availability can restrict metabolic activity, reducing respiration rates. After physical damage, such as grazing or pruning, respiration may increase temporarily as the plant diverts energy toward repair and regrowth. These shifts help allocate energy where it is most needed at any given time. Not all sugars used in respiration come directly from recent photosynthesis. Plants store energy in various forms, often as starch within roots, stems, or specialized storage organs. During periods without sufficient light, such as winter or prolonged shading, these reserves are gradually mobilized. Respiration draws on them to maintain essential functions until photosynthesis can resume at higher levels. The balance between photosynthesis and respiration determines whether a plant is gaining or losing energy overall. When photosynthesis exceeds respiration, surplus sugars accumulate, supporting growth and storage. When respiration exceeds photosynthesis, reserves are depleted. Plants manage this balance continuously, adjusting growth rates and energy use to match conditions. What distinguishes plant respiration from animal respiration is its integration into a slower life strategy. Energy release is steady rather than explosive. There are no bursts of motion or sudden demands. Instead, respiration supports gradual change, allowing plants to grow millimeter by millimeter and maintain internal order over long periods. By pairing photosynthesis with respiration, plants create a closed energy loop. Light energy is captured, stored, released, and redistributed in carefully controlled steps. This loop runs quietly in every living plant, sustaining processes that are rarely visible but always ongoing. Understanding respiration alongside photosynthesis helps complete the picture of plant energy use. It reveals plants not as passive producers of oxygen, but as living systems actively managing energy from moment to moment, day and night, without pause. Across the surface of most leaves are thousands of microscopic openings, known as stomata. Individually, each stoma is too small to see without magnification. Collectively, they form one of the most important regulatory systems in plant life. Through these tiny openings, plants manage a constant trade-off between gaining what they need and losing what they must protect. Stomata serve as gateways between the internal spaces of a leaf and the surrounding air. When they open, carbon dioxide enters the leaf, making it available for photosynthesis. At the same time, oxygen produced inside the leaf can diffuse outward. This exchange is essential for energy production and respiration, but it comes with a cost. Water vapor escapes through the same openings, drifting away into the atmosphere. Because water loss can threaten a plant's stability, stomata are not left open continuously. Each opening is surrounded by a pair of specialized cells called guard cells. These cells change shape in response to internal and external conditions, widening or narrowing the pore between them. When guard cells are full of water, they curve outward, opening the stoma. When they lose water, they relax inward, closing it. This opening and closing is not random. It reflects careful regulation based on multiple signals at once. Light is one of the strongest cues. In many plants, stomata open during the day when light is available for photosynthesis and close as darkness approaches. This timing allows plants to emit carbon dioxide when it can be used efficiently, while limiting water loss during periods when photosynthesis is inactive. Humidity also plays a role. In dry air, water evaporates more rapidly from leaf surfaces. Under these conditions, guard cells may lose water quickly, causing stomata to close partially or completely. This response helps conserve internal water, even if it temporarily limits carbon dioxide intake. In humid air, evaporation slows, allowing stomata to remain open longer without excessive water loss. Temperature influences stomatal behavior as well. Higher temperatures increase evaporation rates, intensifying water loss through open stomata. Plants often respond by adjusting stomatal opening to maintain balance. This regulation helps protect tissues from dehydration, while still supporting essential gas exchange. One of the more subtle aspects of stomatal control is anticipation. Many plants begin opening their stomata before direct sunlight reaches the leaf surface. This behavior is guided by internal circadian rhythms rather than immediate light exposure. By preparing in advance, plants can begin photosynthesis quickly when light becomes available, improving efficiency without waiting for conditions to change. Stomata also respond to internal signals related to water availability within the plant. When soil moisture is low, roots generate chemical signals that travel upward, influencing guard cell behavior in leaves. In response, stomata may close even if light levels remain high. This coordination between roots and leaves allows the entire plant to respond to drought as a unified system. The distribution of stomata varies among plant species and environments. Some plants have stomata primarily on the underside of leaves, reducing direct exposure to sunlight and heat. Others, especially those adapted to aquatic or shaded environments, distribute stomata more evenly. Desert plants often show specialized arrangements that limit water loss, sometimes opening stomata primarily at night. This nighttime opening, seen in certain plants adapted to arid conditions, reflects an alternative strategy for managing exchange. By opening stomata during cooler, more humid nighttime hours, these plants reduce water loss while still taking in carbon dioxide. The carbon dioxide is stored temporarily and used for photosynthesis during daylight hours. This approach highlights how stomatal regulation can be tuned to extreme environments. Despite their small size, stomata have large-scale effects. The combined water vapor released through stomata contributes significantly to local humidity and influences regional climate patterns. Forests, in particular, release vast amounts of water into the atmosphere through this process, shaping rainfall and temperature over broad areas. At the level of the individual plant, stomata embody restraint rather than excess. They do not remain open simply because resources are available. Instead, they adjust constantly, opening just enough and closing just in time. Through this careful regulation, plants maintain internal balance while remaining connected to the air around them. In the quiet opening and closing of these microscopic pores, plants manage survival one breath at a time, sustaining life through measured exchange rather than force or urgency. Once sugars are produced in the leaves, they must be moved to other parts of the plant. Leaves are sites of energy capture, but they are not the only tissues that require energy. Roots, stems, developing buds, flowers, and seeds all depend on a steady supply of carbohydrates and chemical signals to function properly. The system responsible for this internal distribution is the phloem. Phloem tissue forms a network of living cells that extends throughout the plant, connecting areas where sugars are produced with areas where they are needed. Unlike xylem, which transports water upward through non-living tubes, phloem relies on active processes within living cells. This allows transport to occur in multiple directions, depending on the plant's needs at any given time. Sugars enter the phloem in regions known as sources. In most plants, mature leaves act as primary sources because they produce sugars through photosynthesis. These sugars are loaded into phloem cells, increasing the concentration of dissolved substances. Water then enters the phloem by osmosis from nearby xylem, creating internal pressure. This pressure drives the movement of sugar-rich fluid through the phloem toward regions called sinks. Sinks are tissues that consume or store sugars rather than produce them. Growing roots, developing fruits, seeds, and new leaves all act as sinks at different times. The direction of flow depends on the relative positions of sources and sinks, not on gravity or fixed pathways. Because source and sink relationships change over time, phloem transport is dynamic. A young leaf may begin as a sink, relying on sugars from older leaves. As it matures and begins photosynthesizing, it becomes a source, exporting sugars to other parts of the plant. This flexibility allows the plant to allocate energy efficiently as it grows and develops. Phloem transport supports more than just energy distribution. Along with sugars, phloem carries amino acids, hormones, and signaling molecules. These chemical messengers coordinate growth, development, and responses to environmental stress. Signals generated in one part of the plant can influence activity elsewhere, creating a level of integration without a central nervous system. For example, when a leaf is damaged by herbivores, chemical signals can move through the phloem to undamaged tissues, triggering increased production of defensive compounds. During drought, signals from roots influence leaf behavior, adjusting growth and resource use. These responses unfold gradually, guided by chemical gradients rather than rapid electrical impulses. The structure of phloem reflects its role as a living transport system. Phloem cells are arranged end to end, forming long channels known as sieve tubes. These tubes lack many internal structures, reducing resistance to flow. Companion cells adjacent to sieve tubes provide metabolic support, maintaining the pressure gradients that drive movement. Energy is required to load and unload sugars at source and sink regions. This investment allows the plant to move resources precisely where they are needed, even against concentration gradients. Unlike passive diffusion, phloem transport is directed and responsive, adapting continuously to the plant's internal state. The speed of phloem transport is modest compared to animal circulatory systems, but it is well matched to plant life. Sugars move steadily, supplying tissues without sudden surges. This consistency supports gradual growth and long-term stability, rather than rapid bursts of activity. Phloem also contributes to seasonal rhythms. In spring, stored carbohydrates are mobilized from roots and stems to support new growth. In autumn, sugars produced by leaves are redirected into storage tissues as leaves prepare to be shed. These shifts reflect changing source-sink relationships as the plant responds to its environment. Through the coordinated movement of sugars and signals, the phloem connects the plant into a functional whole. Each part remains aware, in a chemical sense, of the needs and conditions of the others. This internal communication allows plants to grow, respond, and persist as integrated systems, guided not by speed or sensation, but by steady distribution and balance. Plant growth does not occur everywhere at once. Unlike animals, whose bodies grow by expanding tissues throughout, plants grow in specific, localized regions. These regions are known as meristems, and they are the quiet centers of construction within a plant. All visible growth, longer roots, taller stems, thicker trunks, can be traced back to the steady activity of these small zones of dividing cells. Meristems are composed of cells that retain the ability to divide repeatedly. While most plant cells eventually specialize and stop dividing, meristematic cells remain in a flexible state. They can produce new cells that either continue dividing or begin to differentiate into specialized tissues, such as vascular cells, epidermal cells, or structural support cells. This ongoing division allows plants to grow throughout their lives, rather than reaching a fixed adult size. There are several types of meristems, each responsible for a different pattern of growth. Apical meristems are found at the tips of roots and shoots. In roots, the apical meristem produces new cells that push the root tip deeper into the soil. In shoots, it extends stems upward and outward, allowing the plant to explore space above ground. These growing tips are protected by surrounding tissues that shield delicate dividing cells from damage. Growth driven by apical meristems is incremental. Cells divide, elongate, and gradually take on specific roles. The plant does not suddenly become taller or longer. Instead, it changes almost imperceptibly, adding new material in small amounts over extended periods. This gradual process aligns with the plant's overall strategy of steady adjustment, rather than rapid transformation. Lateral meristems are responsible for growth in thickness. In woody plants, the most important lateral meristem is the vascular cambium. This thin layer of dividing cells lies between xylem and phloem tissues and produces new layers of each as time passes. As new xylem accumulates on the inside, stems and trunks become wider and stronger. This slow thickening allows plants to support increasing weight and resist mechanical stress. Another lateral meristem, the cork cambium, produces protective outer layers. As woody stems expand, outer tissues may crack or lose function. The cork cambium generates new protective cells that form bark, shielding internal tissues from dehydration, pathogens, and physical injury. This replacement process happens quietly, maintaining protection as the plant grows larger. One of the most distinctive features of plant growth is that old tissues remain in place while new tissues are added. A tree does not replace its trunk each year. Instead, it adds layers to what already exists. Leaves may be shed and replaced, but stems and roots often persist for decades or longer. This layered construction allows plants to record their own history within their structure. Because growth is localized, damage does not necessarily halt development. If a growing tip is lost, nearby meristems or dormant buds may become active, redirecting growth in new directions. This flexibility allows plants to recover from grazing, pruning, or environmental stress without rebuilding their entire structure. Meristem activity is influenced by internal signals and environmental conditions. Temperature, water availability, and nutrient supply all affect how quickly cells divide and differentiate. During favorable conditions, growth may proceed steadily. During unfavorable periods, division slows or pauses, conserving resources until conditions improve. What makes meristems especially suited to plant life is their patience. They do not rush construction or respond impulsively. Growth unfolds cell by cell, layer by layer, guided by local conditions and chemical signals rather than centralized control. This approach allows plants to remain adaptable while maintaining structural integrity over long spans of time. By concentrating growth in specific regions, plants avoid the need for constant restructuring. Stability and change coexist. The plant remains anchored in place, yet it is never finished. New tissues continue to form, extending roots, lifting leaves, and thickening stems as long as conditions allow. Meristems make it possible for plants to grow without movement, to change without rearranging what already exists. Through their quiet, continuous activity, they shape the plant's form over time, building living structures that persist through seasons, disturbances, and years of slow accumulation. Plants do not make decisions in the way animals do. They have no nervous system to process information and no brain to issue commands. Yet plants grow in organized, purposeful ways. Stems bend toward light. Roots grow downward into soil. Buds form where they are most useful. These coordinated patterns are guided not by intention, but by chemistry. Plant hormones are small molecules that influence how cells grow, divide, and specialize. They move through plant tissues in low concentrations, triggering changes in behavior depending on where and when they are present. Rather than acting as on-off switches, plant hormones adjust rates and directions, fine-tuning growth across the plant. One of the most studied plant hormones is auxin. Auxin plays a central role in directional growth. In stems, auxin tends to accumulate on the shaded side when light comes from one direction. This uneven distribution causes cells on that side to elongate more than those on the illuminated side. As a result, the stem bends toward the light source. The movement is slow and subtle, but over time, it reorients the plant to improve light capture. Auxin also helps roots respond to gravity. In roots, higher concentrations of auxin inhibit elongation rather than promote it. When gravity causes auxin to accumulate on one side of a root, growth slows on that side while continuing on the opposite side, causing the root to curve downward. This difference in response between roots and shoots allows a single chemical signal to guide opposite behaviors in different tissues. Gibberellins are another group of hormones that influence growth, particularly elongation. They promote cell expansion and are involved in processes such as stem lengthening, seed germination, and flowering. When gibberellin levels are high, stems tend to grow taller and internodes lengthen. Changes in gibberellin activity can dramatically alter plant form, producing compact or elongated growth depending on conditions. Cytokinins influence cell division and aging. They are often produced in roots and transported upward through the plant. Cytokinins promote the division of cells in growing tissues and delay the aging or senescence of leaves. By balancing the effects of other hormones, cytokinins help regulate how long tissues remain active and productive. Plant growth is rarely controlled by a single hormone acting alone. Instead, hormones interact with one another, creating gradients and balances that shape development. a slight increase in one hormone relative to another can shift growth patterns, redirect resources, or alter the timing of developmental events. This interplay allows plants to respond flexibly to environmental conditions. Hormonal signals move through plants in several ways. Some travel through vascular tissues such as xylem and phloem, reaching distant regions. Others move from cell to cell through direct connections. Because transport is relatively slow, responses unfold gradually. This pace suits plant life, where change is measured in hours, days, or seasons rather than seconds. Environmental cues influence hormone production and distribution. Light, temperature, mechanical stress, and nutrient availability all affect hormonal balance. A plant growing in shade produces different hormone patterns than one growing in full sun. A plant experiencing drought may adjust hormone levels to slow growth and conserve resources. Through hormones, plants maintain coordination without centralized control. Each region responds to local chemical conditions, and the overall pattern of growth emerges from countless small adjustments. There is no single signal directing the entire plant, yet the result is a coherent form that adapts continuously to its environment. By replacing the idea of decision-making with chemistry, hormonal regulation reveals how plants achieve complex behavior through simple principles. Gradients, concentrations, and sensitivity guide growth and form. Direction emerges not from thought, but from balance. In this way, plants remain responsive without haste. Their growth reflects ongoing chemical conversation rather than rapid reaction, allowing them to adjust quietly and persist within the constraints of a rooted life. Plants do not react in sudden ways. Their responses are not immediate, dramatic, or easily noticed. Instead, they unfold gradually, shaped by physical forces and chemical signals acting over time. Even so, plants are constantly responding to their surroundings. Light, gravity, and physical contact all influence how a plant grows, where it directs its energy, and how it positions itself within its environment. One of the most familiar plant responses is phototropism, the tendency of stems and leaves to grow toward light. This response helps plants place their photosynthetic surfaces where light is most available. When light reaches a plant unevenly, chemical signals redistribute growth hormones within the stem. Cells on the shaded side elongate slightly more than those on the illuminated side, causing the stem to curve. The movement is slow and often imperceptible in real time, but over hours or days, the change becomes clear. Phototropism is not about chasing light in a conscious sense. It is a physical outcome of uneven growth. Each cell responds locally to hormone concentrations and light exposure. Together, these tiny differences add up to a visible shift in direction. This allows plants to adjust their orientation continuously as light conditions change throughout the day or across seasons. Gravity exerts an equally constant influence. Plants grow in a world where up and down matter, and they must orient themselves accordingly. Gravitropism refers to growth in response to gravity. Roots typically grow downward, anchoring the plant and accessing water and nutrients, while shoots grow upward, lifting leaves toward light and air. Gravity sensing occurs within specialized cells that contain dense particles. These particles settle in response to gravity, providing positional information. This information influences hormone distribution, which in turn affects growth rates on different sides of the organ. In roots, this results in downward curvature. In shoots, it produces upward growth. As with phototropism, the response is gradual, relying on differential growth rather than movement. Touch also influences plant growth. Thigmotropism describes growth in response to physical contact. Vines and climbing plants exhibit this clearly. When a tendril brushes against a support, contact triggers changes in growth on different sides of the structure. Cells on the side touching the support slow their expansion, while cells on the opposite side continue to grow. Over time, the tendril wraps around the object, securing the plant in place. This response allows climbing plants to take advantage of surrounding structures without investing energy in thick, self-supporting stems. By relying on contact cues, they can rise toward light using existing supports. The response is precise but unhurried, unfolding over hours as growth patterns adjust. Plants also respond to mechanical stress more broadly. Repeated exposure to wind or movement can alter growth form. Stems may become shorter and thicker, increasing stability. This phenomenon reflects the plant's ability to sense strain and adjust structure accordingly. The result is not avoidance, but reinforcement, strengthening tissues where stress is consistently applied. These responses do not require awareness or interpretation. They emerge from physical interactions between cells, hormones, and environmental forces. Light, gravity, and touch influence chemical gradients, and those gradients guide growth. The plant does not decide where to grow. It responds through the laws of physics and chemistry, acting within living tissue. The slow pace of these responses offers advantages. Gradual change reduces the risk of overcorrection. It allows plants to integrate multiple signals over time, rather than reacting to brief fluctuations. A momentary shadow or a passing touch does not immediately redirect growth. Instead, sustained conditions guide long-term form. Together, these responses help plants occupy space efficiently. Light guides leaves upward and outward. Gravity anchors roots and stabilizes structure. Touch allows plants to climb, brace, or strengthen themselves against movement. Each response increases survival without requiring movement from place to place. In this way, plants demonstrate a form of responsiveness that is steady rather than reactive. Their growth reflects continuous adjustment to the forces acting upon them. Over time, these adjustments shape the plant's form, allowing it to remain rooted while still responding meaningfully to the world around it. Plants are rooted in place. They cannot flee from insects, grazing animals, or pathogens that threaten to consume or damage them. Instead, plants rely on chemical strategies that reduce the likelihood of being eaten or infected. These chemical defenses are not passive byproducts of metabolism. They are active components of survival, produced, regulated, and adjusted in response to risk. Many plants synthesize compounds known as secondary metabolites. Unlike sugars or proteins, which are essential for basic cellular function, secondary metabolites serve specialized roles. A large number of these compounds function as deterrents, making plant tissues toxic, bitter, irritating, or otherwise undesirable to potential consumers. Through chemistry, plants influence the behavior of organisms around them. Alkaloids are one major class of defensive compounds. These nitrogen-containing molecules often interfere with the nervous systems of insects and other animals. Nicotine, caffeine, and morphine are all alkaloids produced by plants. In small doses, these substances may discourage feeding or reduce insect activity. In larger amounts, they can be lethal. For the plant, producing alkaloids creates a chemical barrier that reduces repeated attacks. Tannins are another group of defensive chemicals. They bind to proteins, reducing their digestibility. When an herbivore consumes leaves rich in tannins, digestion becomes less efficient, and nutrient absorption declines. Over time, this discourages feeding on tannin-rich plants. Tannins also have antimicrobial properties, helping protect damaged tissues from infection. Phenolic compounds, which include a wide range of molecules, contribute to both defense and structural support. Some phenolics act as toxins or feeding deterrents. Others strengthen cell walls, making tissues tougher and less palatable. Together, these compounds increase the cost of consumption for herbivores and pathogens. Chemical defenses are not always present at constant levels. Many plants increase production after damage occurs. When leaves are chewed or stems are wounded, internal signaling pathways activate enzymes responsible for synthesizing defensive compounds. This allows the plant to respond proportionally, investing energy in defense when risk is high and conserving resources when it is low. Some defensive chemicals serve multiple purposes. Capsaicin, the compound responsible for the heat of chili peppers, deters mammals while having little effect on birds, which help disperse seeds. In this way, chemical defense can be selective, shaping interactions with different species rather than simply repelling all consumers. The production of defensive chemicals requires energy and resources. As a result, plants balance defense with growth. Under favorable conditions, when light and nutrients are abundant, plants may invest more in chemical protection. Under stressful conditions, growth and maintenance may take priority. This balance shifts continually, guided by internal signals and environmental cues. Chemical defenses influence entire ecosystems. Herbivores adapt to specific plant toxins, sometimes evolving resistance. In turn, plants may alter their chemical profiles over generations. These interactions drive evolutionary change, shaping the diversity of plant and animal life. From a distance, a plant may appear unguarded, its leaves exposed and still. At the chemical level, however, it may be anything but defenseless. Invisible compounds circulate within tissues, deter feeding, and influence the behavior of surrounding organisms. Chemical defense allows plants to remain rooted, while still exerting control over their interactions. Rather than relying on movement or aggression, they rely on chemistry and time. These strategies unfold quietly, shaping survival outcomes without drawing attention, reinforcing the idea that stillness does not imply vulnerability. Alongside chemical deterrents, plants rely on physical structures to reduce damage from herbivores, environmental stress, and infection. These physical defenses are built into the plant's form, shaping how it interacts with the world. Unlike chemical defenses, which can change rapidly, structural barriers represent long-term investments made over the course of growth. One of the most familiar physical defenses is the thorn or spine. Thorns are modified stems or branches, while spines are often derived from leaves or parts of leaves. Both serve a similar function, discouraging large herbivores from feeding. By creating sharp, rigid projections, plants increase the risk of injury to animals that attempt to browse on their tissues. Over time, this encourages animals to seek food elsewhere. Smaller physical defenses also play important roles. Trichomes are fine hair-like structures that cover the surfaces of leaves and stems in many plants. These structures vary widely in form and function. Some trichomes are soft and reflective, reducing heat and water loss by shading the leaf surface. Others are stiff or hooked, making it difficult for insects to move across the plant. Certain trichomes even release irritating substances when touched, combining physical and chemical defense. The outer surfaces of plants are often coated with a protective layer known as the cuticle. This waxy barrier covers epidermal cells and reduces water loss by limiting evaporation. It also serves as a first line of defense against pathogens and physical damage. The thickness and composition of the cuticle vary depending on the plant's environment. Plants in dry or sunny conditions often develop thicker cuticles to protect against dehydration and ultraviolet radiation. In woody plants, bark provides a more substantial barrier. Bark is composed of multiple layers of dead and living cells that insulate and protect internal tissues. It shields the plant from temperature extremes, mechanical injury, and invasion by insects or microorganisms. As the plant grows, bark is continuously renewed by underlying tissues, maintaining protection even as outer layers crack or peel away. Structural defenses require significant energy and time to build. Producing lignin, thick cell walls, and protective outer layers diverts resources that could otherwise be used for rapid growth. Because of this cost, physical defenses are often most pronounced in long-lived plants that benefit from durable protection over many seasons. The arrangement of tissues within stems and leaves also contributes to defense. Tough fibers and reinforced cell walls make tissues harder to chew and digest. Some plants increase tissue density in response to repeated damage, strengthening areas that experience frequent stress. This ability to reinforce structure over time helps plants adapt to persistent threats. Physical barriers also play a role in disease resistance. By limiting entry points for pathogens, structures such as thick cuticles and bark reduce the likelihood of infection. Even when damage occurs, surrounding tissues can compartmentalize wounds, isolating affected areas and preventing spread. This localized response protects the rest of the plant without requiring wholesale change. Beyond defense, structural features often serve multiple functions. Spines may also reduce water loss by shading surfaces or disrupting airflow. Thick bark can store water or carbohydrates. Trichomes can influence microclimates around leaves. These overlapping roles make physical defenses efficient adaptations rather than single-purpose traits. Through physical defenses, plants shape their interactions with the environment in lasting ways. Their forms reflect past pressures, encoded into structure through growth. These barriers do not react quickly or dramatically, but they persist, providing continuous protection. By investing in structure, plants create boundaries that define where and how interaction occurs. These boundaries allow them to remain exposed to light and air, while reducing the costs of vulnerability. In this balance between openness and protection, physical defenses contribute to the quiet resilience that characterizes plant life. Plants do not exist in isolation. Even when they appear separated by space, they remain connected through the air above them and the soil below. Communication in plants does not involve intention or language, but it does involve information. Through chemical signals, plants influence one another's growth, chemistry, and behavior in ways that increase collective resilience. One form of plant communication occurs through the release of volatile organic compounds, or VOCs. These are small, airborne molecules that easily diffuse through the air. When a plant is damaged by insects or other herbivores, it may release a specific blend of VOCs. These compounds can serve multiple functions at once, depending on context. Some VOCs act directly as deterrents, making the plant less attractive to herbivores. Others function as indirect defenses by attracting predators or parasites of the herbivores. For example, certain plants release compounds that draw in insects which prey on the insects feeding on the plant. This layered response allows plants to influence interactions beyond their immediate tissues. Airborne signals can also affect neighboring plants. Nearby individuals of the same or different species may detect these compounds and adjust their own chemistry in response. This can include increasing the production of defensive chemicals or altering growth patterns. In this way, information about damage spreads through a population without direct contact. Communication also occurs below ground, where roots interact with soil and with each other. Plant roots release a wide range of substances into the surrounding soil, collectively known as root exudates. These include sugars, amino acids, organic acids, and signaling molecules. Root exudates influence microbial communities, shaping which bacteria and fungi thrive nearby. These underground interactions extend beyond microbes. Chemical signals in the soil can affect the growth of neighboring plants, either inhibiting or promoting development. Some plants release compounds that suppress the germination or growth of competitors, a phenomenon known as allelopathy. This chemical influence helps structure plant communities and reduce direct competition for resources. Mycorrhizal fungi play a central role in underground communication. These fungi form extensive networks that connect the roots of multiple plants. Through these networks, resources such as water and nutrients can be shared. Chemical signals associated with stress or damage may also move along these pathways, influencing responses in connected plants. The movement of signals through fungal networks does not imply cooperation in a human sense. Instead, it reflects the interconnected nature of biological systems. Fungi benefit from access to plant sugars, while plants benefit from improved nutrient uptake and communication. These exchanges arise from mutual advantage rather than coordination or planning. Plant communication through air and soil operates on timescales suited to plant life. Signals may take minutes to hours to disperse and trigger responses. These slower dynamics reduce the likelihood of overreaction and allow plants to integrate information from multiple sources. A single damaged leaf does not necessarily trigger a full defensive response across an entire ecosystem. Environmental conditions influence how signals travel. Wind patterns affect the spread of airborne compounds. Soil composition and moisture influence how chemicals diffuse underground. These physical factors shape communication, adding variability and context to signaling processes. What distinguishes plant communication is its subtlety. There are no visible gestures or audible signals. Information moves invisibly, carried by molecules rather than sound or light. Yet the effects can be profound, altering growth, defense, and survival across communities of plants. By communicating through air and soil, plants extend their influence beyond their own tissues. They participate in networks that blur the boundaries between individuals. Each plant remains rooted and separate, yet chemically connected to its surroundings. This form of communication reinforces the idea that plant life is not solitary. Even in apparent stillness, plants exchange information with the living world around them, responding to shared conditions through quiet chemical conversation rather than rapid reaction. Beneath the surface of the soil, plant roots rarely grow alone. Surrounding them is a dense and active world of fungi, bacteria, and microscopic organisms, all interacting continuously. Among the most influential of these relationships are mycorrhizal partnerships, long-term associations between plant roots and fungi that reshape how plants acquire resources and interact with one another. Mycorrhizal fungi attach themselves to plant roots and extend far beyond them into the surrounding soil. These fungal threads, known as hyphae, are extremely thin, often much finer than plant roots themselves. Because of their size and reach, hyphae can explore soil volumes that roots alone cannot access. They move through small pores around soil particles and into spaces that would otherwise remain unreachable. Through this expanded network, fungi absorb water and minerals from the soil, particularly nutrients that are difficult for plants to obtain on their own. Phosphorus is one such nutrient, often present in soil, but locked into forms that roots struggle to access. The fungi mobilize these nutrients and transport them back to the plant. In exchange, the plant supplies the fungi with sugars produced through photosynthesis. These sugars serve as an energy source for the fungi, allowing them to grow, maintain their networks, and continue exploring the soil. This exchange is not symbolic or voluntary. It is a biochemical trade governed by gradients, transport proteins, and long-term mutual benefit. What makes mycorrhizal partnerships especially significant is their scale. A single fungal network can connect the roots of many different plants, sometimes spanning large areas underground. Through these shared connections, resources do not always move evenly. Some plants may receive more nutrients than others, depending on factors such as light availability, root access, and fungal distribution. The network responds to conditions rather than enforcing equality. These connections also allow chemical signals to move between plants. When one plant experiences stress, such as drought or damage, chemical changes associated with that stress can travel through fungal pathways. Neighboring plants connected to the same network may adjust their own physiology in response, altering growth rates, water use, or defensive chemistry. This signaling does not require direct root contact or airborne cues. It occurs entirely below ground, out of sight. The existence of these networks challenges the idea of plants as independent individuals. While each plant maintains its own tissues and boundaries, mycorrhizal connections blur the edges between them. Nutrients, water, and information move through shared pathways, creating systems that function at the level of communities rather than isolated organisms. Forests provide some of the clearest examples of this interconnectedness. Trees of different ages and species are often linked by extensive fungal networks. Young seedlings growing in shade may receive carbon compounds indirectly through these connections, helping them survive until they can access sufficient light. Older trees, with larger photosynthetic capacity, contribute more resources to the network, while receiving mineral support from fungi in return. Grasslands and agricultural soils also rely heavily on mycorrhizal activity. In these environments, fungal networks influence soil structure, water retention, and nutrient cycling. The presence or absence of healthy mycorrhizal communities can significantly affect plant productivity and resilience, even when soil chemistry appears unchanged. These partnerships develop slowly. Fungal networks expand over time, responding to root growth, soil conditions, and seasonal changes. They persist across years, sometimes even decades, adapting as plant communities shift. Disturbance to the soil, such as intensive tilling or compaction, can disrupt these networks, breaking connections that took long periods to form. What makes mycorrhizal systems remarkable is not speed or coordination, but persistence. They operate continuously, redistributing resources and signals without central control. Each interaction is local, governed by immediate conditions, yet the cumulative effect shapes entire ecosystems. From the surface, a field or forest may appear as a collection of separate plants. Below ground, those plants are often linked by shared fungal pathways, exchanging materials and responding collectively to their environment. This hidden cooperation allows plant communities to buffer stress, stabilize soils, and make efficient use of limited resources. By revealing these underground partnerships, we begin to see plant life not as solitary growth, but as participation in long-term networks. These systems remain unseen, yet they quietly support much of the life above them, reinforcing the idea that stillness at the surface often conceals complexity below. Nitrogen is one of the most important elements in plant life. It is a key component of proteins, enzymes, and genetic material, and without it, growth quickly slows or stops. Yet despite being abundant in the atmosphere, nitrogen is not easily accessible to most plants. The vast majority of atmospheric nitrogen exists in a stable form that plants cannot use directly. This limitation has shaped some of the most important partnerships in terrestrial ecosystems. Most plants depend on nitrogen already present in the soil, absorbed in forms such as nitrate or ammonium. These usable forms originate from the activity of microorganisms that convert nitrogen from one chemical state to another. Without this microbial processing, nitrogen would remain largely unavailable, regardless of how much surrounds the plant in the air. Among the most specialized and influential of these processes is nitrogen fixation. Nitrogen-fixing microorganisms possess enzymes capable of breaking the strong bonds that hold atmospheric nitrogen together. Through this process, nitrogen gas is converted into ammonia, a form that can be incorporated into organic molecules. This transformation requires energy and occurs only under specific conditions. which is why it is carried out by relatively few organisms. Some plants form close, long-term relationships with nitrogen-fixing bacteria. Legumes are the most familiar example. Plants such as beans, peas, clover, and alfalfa develop specialized structures on their roots called nodules. Within these nodules live bacteria that perform nitrogen fixation. The plant provides carbohydrates and a protected environment, while the bacteria supply nitrogen in a usable form. Inside the root nodules, conditions are carefully regulated. Nitrogen fixation enzymes are sensitive to oxygen, yet the bacteria require oxygen for respiration. The plant manages this balance by controlling oxygen levels within the nodule, allowing fixation to proceed efficiently without damaging the enzymes involved. This precise regulation reflects the depth of integration between plant and microbe. The nitrogen provided through this partnership is incorporated into amino acids and other nitrogen-containing compounds. These molecules support growth, repair, and reproduction. Because the nitrogen is supplied directly to the plant, legumes can thrive in soils that are otherwise low in available nitrogen. This gives them a competitive advantage in certain environments and allows them to colonize nutrient-poor areas. Nitrogen-fixing partnerships have effects beyond individual plants. When legume tissues die and decompose, nitrogen is released into the soil, becoming available to other plants. In this way, nitrogen fixation enriches ecosystems over time, increasing overall fertility and supporting greater plant diversity. Not all nitrogen-fixing bacteria live inside plant roots. Some exist freely in the soil or associate loosely with plant surfaces. These microbes also contribute to the gradual buildup of usable nitrogen, though their effects are often more diffuse. Together, these microbial processes form the foundation of nitrogen cycling in terrestrial environments. Agricultural systems rely heavily on nitrogen availability. Modern farming often supplements soil nitrogen with synthetic fertilizers, but biological nitrogen fixation remains a critical natural input. In many farming practices, legumes are intentionally included in crop rotations to replenish soil nitrogen and reduce the need for external inputs. This approach mirrors processes that occur naturally in unmanaged ecosystems. Nitrogen fixation is energetically costly. Both plants and bacteria must invest resources to maintain these partnerships. As a result, nitrogen-fixing relationships tend to form only when the benefits outweigh the costs, such as in nitrogen-poor soils. When nitrogen is abundant, plants may reduce investment in nodules or rely more heavily on direct soil uptake. The dependence of plants on microbial allies highlights an important theme in plant life. Access to resources often depends on relationships rather than individual ability. Roots alone cannot unlock atmospheric nitrogen. It is only through cooperation with specialized microbes that this essential element becomes available. These microbial processes operate continuously, largely unnoticed. Over time, they shape soil fertility, plant productivity, and ecosystem stability. Forests, grasslands, and agricultural fields all depend on nitrogen fixation to sustain growth across seasons and generations. By understanding nitrogen fixation, we see how plant nutrition extends beyond soil particles and root hairs. It reaches into the atmosphere and into the microscopic world of bacteria, linking plants to global chemical cycles. Plants experience time differently than animals do. Their lives unfold at speeds that are often too slow to notice in daily observation, yet they are deeply shaped by time at multiple scales. From brief life cycles lasting only weeks to lifespans that stretch across millennia, plants embody a wide range of temporal strategies, each suited to specific environments and conditions. Some plants complete their entire life cycle in a single growing season. These annual plants germinate, grow, reproduce, and die within months. Their strategy emphasizes speed and reproduction, allowing them to take advantage of short windows of favorable conditions. By producing large numbers of seeds, they ensure that future generations can persist, even if individual plants are short-lived. Other plants adopt a longer view. Perennials live for multiple years, maintaining roots or stems that survive across seasons. Growth may slow or pause during unfavorable conditions, but the plant remains alive, ready to resume activity when circumstances improve. This approach allows plants to invest in stronger structures and more stable resource storage. At the extreme end of the spectrum are plants that live for hundreds or even thousands of years. Certain trees persist through centuries of environmental change, recording their history in annual growth rings. These long-lived plants grow slowly, investing in durability and resistance rather than rapid expansion. Their continued survival reflects a balance between growth, repair, and defense maintained over long periods. Growth rates in plants are closely tied to environmental stability. In predictable climates with consistent resources, plants may grow steadily year after year. In more variable environments, growth may be episodic, accelerating during favorable periods and slowing dramatically during stress. These patterns reflect an ongoing negotiation between opportunity and restraint. Seasonal cycles impose powerful rhythms on plant life. Changes in temperature, day length, and water availability signal when to grow, reproduce, or enter dormancy. In temperate regions, many plants reduce metabolic activity during winter, conserving energy and protecting sensitive tissues. As conditions improve, growth resumes, often rapidly at first, drawing on stored reserves. Beyond seasonal changes, plants also operate on daily cycles. Circadian rhythms influence leaf movement, stomatal behavior, hormone production, and metabolic activity. These internal clocks allow plants to anticipate regular changes in light and temperature, rather than responding only after conditions shift. By aligning internal processes with external cycles, plants improve efficiency and reduce stress. Leaf movements provide a visible example of daily rhythms. Some plants adjust leaf angles throughout the day, optimizing light capture in the morning and reducing exposure during intense midday sunlight. Others fold or reposition leaves at night, a behavior linked to changes in internal pressure and circadian regulation. These movements are subtle but consistent, reflecting the plant's ongoing awareness of time. Chemical processes within plants also follow rhythmic patterns. Photosynthesis, respiration, and hormone production fluctuate over the course of the day. These fluctuations help balance energy capture with water conservation and repair. Even when environmental conditions remain constant, internal rhythms persist, guiding timing and coordination. Plants respond not only to immediate conditions, but also to accumulated experience over time. Prolonged drought, repeated disturbance, or long-term climate trends can alter growth patterns and developmental timing. These changes reflect plasticity rather than memory in the human sense, yet they allow plants to adjust strategies across seasons and years. By spanning such a wide range of lifespans and rhythms, plants demonstrate that time in biology is not uniform. Life can move quickly or slowly, pause and resume, or persist almost unchanged across generations. What matters is alignment with environment, not speed. Understanding plant time helps reframe stillness. A plant standing quietly in one place is not inactive. It is synchronized with cycles that extend beyond immediate perception, responding to light, temperature, and season with patience rather than urgency. Through their varied lifespans and rhythms, plants reveal that life does not require constant motion to remain dynamic. Human life is inseparable from plant life. Long before modern cities, technologies, or written records, plants shaped where humans could live, what they could eat, and how societies developed. Even today, surrounded by artificial structures and controlled environments, human systems remain deeply dependent on plant biology. Agriculture is the most direct example of this dependence. Crops provide the majority of the calories consumed by humans, either directly through plant foods or indirectly through animals that feed on plants. The success of agriculture depends on understanding plant growth, water needs, nutrient requirements, and seasonal timing. Farmers work within the limits set by plant physiology, adjusting practices to align with how plants respond to light, temperature, and soil conditions. Over thousands of years, humans have altered plants through domestication. By selecting individuals with desirable traits such as larger seeds, sweeter fruits, or reduced bitterness, humans gradually changed plant populations. These changes often came with trade-offs. Many domesticated plants are less able to survive without human care, relying on irrigation, protection, and nutrient management. This mutual dependence reflects a long coevolution between humans and plants. Forests have also played a central role in human history. Trees provide building materials, fuel, and shelter. They regulate local climates, stabilize soils, and influence water cycles. The presence or absence of forested land has shaped settlement patterns, economies, and cultural practices. Even today, forest health affects air quality, water availability, and biodiversity at regional and global scales. Gardens represent a more intimate human-plant relationship. Whether ornamental or functional, gardens reflect deliberate interaction with plant growth. Pruning, watering, and planting schedules are guided by plant responses, rather than human convenience alone. Gardeners learn to observe slow changes, recognizing signs of stress, growth, or dormancy that unfold over weeks and months. Indoor plants bring plant life into controlled environments, yet they do not cease to follow biological rhythms. Even inside buildings, plants respond to light cycles, temperature changes, and water availability. Leaves orient toward windows. Growth slows in low light. Dormancy may occur despite constant indoor conditions. These responses reflect internal regulation shaped by evolutionary history, rather than immediate surroundings. Urban environments pose unique challenges for plants. Compacted soils, limited root space, air pollution, and altered temperature patterns influence growth and survival. Some species adapt well to these conditions, while others struggle. The plants that persist in cities often display resilience, tolerating stress and variability while continuing to provide ecological benefits such as cooling, shade, and habitat. Human attempts to control plant growth often reveal the limits of intervention. Overwatering, excessive fertilization, or inappropriate lighting can disrupt internal balances, leading to decline rather than improvement. Successful cultivation depends on aligning care with plant biology rather than overriding it. This alignment requires patience and observation rather than force. Plants also shape human perception of time and space. Seasonal changes in vegetation mark the passage of time, signaling planting and harvest, growth and rest. Landscapes defined by plant communities influence how humans experience place, from open grasslands to dense forests. These influences operate subtly, shaping behavior and culture without direct instruction. Through crops, forests, gardens, and indoor spaces, plants remain embedded in human life. They respond to human actions, but they are not fully controlled by them. Their needs, rhythms, and limits continue to assert themselves, reminding humans that built environments rest on biological foundations. By recognizing plants as active participants in human environments, it becomes clear that human systems do not stand apart from nature. They are layered on top of it, dependent on plant processes that continue quietly beneath daily routines. Even in the most developed settings, plant life persists, shaping outcomes through slow, steady influence rather than immediate change. As we come to rest at the end of this exploration, it may help to let the idea of plants settle quietly in the background of your thoughts. Plants do not hurry. They do not pause to announce what they are doing. They grow, exchange, respond, and persist without drawing attention to themselves. Their lives unfold alongside ours, shaped by light, water, chemistry, and time, following rhythms that are steady rather than urgent. Everything we've explored points to a simple truth. Stillness does not mean inactivity. Beneath the appearance of calm, plants are constantly maintaining balance. Water continues to move upward through stems. Sugars are redistributed. Cells divide slowly at growing tips. Chemical signals drift through air and soil. These processes continue whether or not they are observed. Plants also remind us that life does not require speed to be effective. Some complete their lives quickly, while others remain rooted in one place for centuries. Some respond within hours, others across seasons. Each strategy is shaped by environment rather than ambition, by patience rather than pressure. When we begin to notice plants as living systems rather than background objects, the world itself can feel quieter and more grounded. Streets lined with trees become places of ongoing exchange. Gardens become slow conversations between soil, light, and growth. Even a single leaf becomes a site of continuous movement, balancing water loss, gas exchange, and energy capture moment by moment. There is comfort in this kind of persistence. Plants endure change without resisting it. They adjust gradually, conserving resources, responding proportionally, and remaining present through conditions that come and go. They remind us that stability often comes not from control, but from alignment with the environment. As you rest now, you might imagine the plants around you, outside, nearby, or far away, continuing their quiet work. Roots extending into soil, leaves releasing water into the air, networks beneath the surface redistributing nutrients and signals. None of it demands attention. None of it requires effort from you. The world remains alive even when it feels still. And as your own thoughts begin to slow, you can let that awareness fade gently into the background, just as plants do, present, steady, and quietly at rest.